Hi dear students, welcome to Dental Amaze Academy. Today we are going to start a very crucial subject which is microbiology, and hopefully for most of you it's your initial phase of your preparation. We are going to start with microbiology. Now you heard me saying that we are starting a very crucial subject. I didn't say important. Why is it? Now, important means you get lots of questions from that particular subject. Crucial means, yes, this microbiology is one subject which can bring down your rank a lot. Bring down in the sense you can get a much, or you can score well with microbiology provided you understand the basics and concepts in microbiology. Okay, now considering NEET, this microbiology you will get around 7 questions as per the marking system. So compared to many clinical subjects and even subjects like general medicine or general surgery, you get half of the number of questions. For example, in all the clinical subjects you get around 14 to 15 questions. General medicine you get around 14 to 15 questions, but here you can expect only around like somewhere around 7 questions. Okay, but yes, this is a very crucial subject, because lots of basics come in here. Subjects like topics like immunity is being taught in the microbiology. You have to understand what are lymphocytes, what are neutrophils, what are different types of immune cells, what are different types of immunity. So all this comes under here. Hypersensitivity reactions, different types of hypersensitivity reactions, the cells involved there. So all these are basic things. And yes, bacteriology. Basic bacteriology and systemic bacteriology. See, we are starting with systemic bacteriology because there is a very related topic which is the basic bacteriology. The problem to start with basic bacteriology is that it's really boring. You just need to, like in basic bacteriology, you study about lots of staining reactions, lots of culture medias and all those, which it's all about by hearting. So instead of going that way, let us study some bacteria, importance of that bacteria, and what is the clinical significance of doing all these culture medias and staining reactions. Then once you go back and learn them, the basic bacteriology, it will be much more, or it will make much more sense. Definitely learning microbiology requires lots of memorizing, but once you understand the concepts, it becomes much more easy. Okay, and another chapter which is very important in microbiology is sterilization. So if you consider AIMS or currently the INI-CET, you see that one of the most important chapters for AIMS is sterilization and disinfection, and bacteriology and viral infections. Now, this bacteriology, viral infection, sterilization can be included under any medical subject, including OMFS. Okay, they can ask the questions from any, like they can include these questions of infections under oral pathology, they can include it under OMFS, okay, because there are space infections involved in the maxillofacial region. So, though you get 7 questions from here, the problem is it's very crucial. Okay, you have to understand this to understand lots of concepts out here in your entrance preparation. Now, starting your entrance preparation. Today I was talking to one student, and the student asked me, like, what if I finish the lectures? Okay, what if I finish the lectures you have given to me and I have spare time? So what will I do then? Now see, if you consider learning more and more topics, you will get a lot of time. You have a lot of time. But the key here is, when you are learning for NEET MDS examination, the key is revision. Okay, so whatever extra time you get, revise whatever you have learned previously, especially volatile subjects like microbiology. And please, the first and foremost thing which you have to follow is to go through the lectures, revise your notes, okay, then go through the MCQ books, and then come to your exams. Okay, then only if you want to do something extra, you can do it. So what does it mean to do something extra? So if you want to solve some extra MCQs, or if you want to read textbooks, all comes after once you have covered your basics. Just see into your previous, ask your seniors about your NEET exam which just got over. It was just the basic things. Okay, there were new things, there were new questions, but majority of the questions were based on simple, direct, basic things. Doesn't mean that the exam is very simple. You have to learn 19 subjects in the coming days. You have to revise it multiple times. So that's the key. Okay, so let's start. In between, we'll discuss and go through more and more details as we go on. Okay, so as I've told you, we are going to start with systemic bacteriology. Now, to start with, I'm going to introduce you to a very important molecule which you will have to learn in microbiology, which is catalase. Very important molecule. Now, what is the significance of this molecule in our body? That's what I'm going to tell you. And then I'll tell you what's the importance of catalase in bacteriology. Okay, so what catalase does? So it's there in almost all living organisms. And what it does is it converts the hydrogen peroxide into water and oxygen. Now, what is the importance of this reaction? Now, hydrogen peroxide in the body is formed due to various reactions or various metabolic reactions taking place in our body. This you will learn in detail about it when we come on to biochemistry and general pathology. But as of now, just understand that this molecule is formed in the body, and this can cause oxidative stress. Okay, this can cause oxidative stress in the body. Means this can oxidize different molecules in the body, and oxidation of molecules can lead to formation of free radicals. Okay, and these free radicals can cause various injuries, inflammations, including cancer. Okay, so this reaction is very crucial, and thus catalase is a very crucial molecule of the body. Now, when this reaction, what we need to know here is when this reaction takes place, what you get is bubbling. Okay, you get bubbling. Okay, so if this catalase reacts with hydrogen peroxide, bubbling will be seen. So that is what we are going to see next. What is the importance of this bubbling? Now you know what is the importance of catalase. Now, this reaction is not very crucial. What is important for us is we have got two groups of bacteria with us. One is Staphylococci, the other is Streptococci. Okay, and we don't know who has got, or you have got, just imagine you have got two dishes. One you have got a particular bacteria, the other you have got another particular bacteria. Okay, now what we are going to do is we are going to put each one of them into catalase. Into hydrogen peroxide, or you have got two culture plates. Okay, you will study what is culture plates and all. Just imagine it's a plate. Okay, you're going to put the hydrogen peroxide into the first culture plate and the second culture plate, and you're going to see which one produces bubbling. And you will notice that this one, okay, this Staphylococci produces bubbling. Means what? What does it mean? It means that Staphylococci has got catalase with them. Streptococci doesn't have catalase in their cell. Okay, so means Staphylococci, the most important, most crucial point is Staphylococci are catalase positive. Okay, Staphylococci are catalase positive organisms. These are catalase negative organisms. We have just begun entrance preparation now, because they're going to ask you directly. At times they can ask you directly, which of the following are catalase positive? Staphylococci, Streptococci, okay, Gonococci, whatever they'll ask you, bacilli, and the answer is Staphylococci. Or they can ask you, a patient came to you with this and this symptom, okay, which you'll learn very soon what are the symptoms, and the organism was found to be catalase positive, coagulase positive. Okay, so what is the organism? Identify the organism. Okay, now you see, you feel like it's so simple. Now you're going to study 20 subjects in the coming 1 year. So that is where you get confused. That is where the confusion starts. Is it Staphylococci or Streptococci? Remember, it's Staphylococci, which is Catalase. Remember, we are going to study Coagulase next. So remember, it's A, C, A comes first. So Catalase is Staphylococci. Okay? Now, the reaction doesn't stop there. Forget about Streptococci now. We are going to concentrate over Staphylococci. Now, this Staphylococci, a group of, or a particular type of Staphylococci has got a particular enzyme known as coagulase. We'll study this coagulase in detail now, okay, after some time. So now we are first classifying the organisms. So what you're going to see is that you're going to add in the Staphylococci group itself. You've got different Staphylococci. You have separate culture medias. What you're going to do is you're going to add some like sheep blood into each cultures. Okay, sheep blood. Just imagine it like that. So each culture you add some blood, sheep blood or blood, just blood. Okay, and you're going to see the reaction. If there is a plasma clot formed, we'll study this in detail, don't worry. If there is a plasma clot formed, you call those organisms as coagulase positive. Okay? If there is no plasma clot formed, you call these organisms as coagulase negative. Coagulase, okay? Or they are named as CONS, coagulase negative, CONS, coagulase negative Staphylococci. Okay, so this is coagulase positive, this is coagulase negative. Now let us see which is the organism which is coagulase positive, or which is the organism that contains the enzyme called coagulase. The organism is Staph. We are discussing about Staph only. The organism is Staph aureus. Okay, Staph aureus. Okay, now that's the most important organism in Staphylococci, okay, which we're going to start with. But before going on to any further, let's understand a little bit on CONS also. What you're going to do then? So there are multiple, now you have got a set of organisms which are CONS organisms or coagulase negative organisms. So we have to identify different coagulase negative organisms. So what we're going to do is we're going to add an antibiotic known as novobiocin. You're going to add novobiocin into this culture, and let us see what is the reaction which is going to take place. Okay, now novobiocin is an antibiotic, so some bacteria will die, some will be alive. So those organisms which die are known as the novobiocin sensitive organisms. Okay, novobiocin sensitive organisms. So one side you have got novobiocin sensitive organism, okay, sensitive, and on the other side you have got novobiocin resistant organisms. Okay, so what are sensitive, novobiocin sensitive, what are novobiocin resistant? That's what we are going to see next. Okay, which are the organisms which come under each group. Novobiocin sensitive organisms are Staph epidermidis, okay, and Staph hemolyticus, okay, lyticus. And whereas on the other side, which is resistant, it is Staph saprophyticus. Just no, okay, saprophyticus. Okay, saprophyticus. Now, this chart is very, very, very important. You can get hundreds of questions, or you can make hundreds of questions from here. Okay, an organism, like a patient came with this and the symptoms, definitely all will have fever and all those. Okay, and on culture or on microbiological examination, whatever, you find that organism is catalase positive, coagulase negative, novobiocin resistant. Which organism? Staph saprophyticus. So anyway, you can get the questions here. Okay, so very important. So catalase positive is Staphylococci, catalase negative is Streptococci. So then you check for Coagulase positivity. You have got positive is Staph Aureus, negative again we divide into based on novobiasm sensitivity. So those are sensitive are epidermidis and hemolyticus. Those are resistant are Staph saprophyticus. Okay. Now let us come on to more details of this Staph aureus. And please note, in general, all Staphylococci, including aureus, okay, has got a particular morphology, which is they are gram positive. So we'll study about gram positivity, gram staining, okay, Ziel-Nelson staining and all in basic bacteriology next. But let us see what are the gram positive, what are the gram negative, okay, and what is the significance of that. Then we'll go on to that. Okay, so they are gram positive organisms which occur in grape-like cluster. Okay, grape-like cluster. Okay, grape-like cluster. So you can see here the grape-like cluster kind of look of these organisms there. Okay, so then one very important thing which you have to know is transformation. They get transformed into L forms. They get transformed into L forms. When they are exposed to the antibiotic called penicillin. Okay, when they are exposed to antibiotic called penicillin, they get transformed into L forms. So what are L forms? We're going to study again in basic bacteriology. Don't worry, if I go directly into basic bacteriology, it will become a worse lecture. But what is L form? L form is a cell wall deficient bacteria. Okay, a form of cell wall deficient. There is no cell wall. So basically you all know that majority of the bacteria has got cell walls, except some like Mycoplasma doesn't have it. But when you expose even the cell wall containing bacteria like Staph aureus to penicillin, the cell wall is lost. And what is the role of this penicillins? Do you know it? We'll study it in pharmacology. Basically it acts on cell wall. Okay, so it makes the bacteria cell wall deficient, which is called L form. Okay, L form. Now, why is it called aureus? Because it has got this particular color. That is, aureus means golden yellow color. Okay, so when you just grow it in culture media, we'll see later on, they will show a golden yellow color. Okay, golden yellow color. So three things here. They are gram positive cocci. So in your MCQ book like Dental Pulse, you will see chart showing gram positive cocci. Like big chart is there. Don't worry. Just understand each and every bacteria. We're going to discuss each and every bacteria in that list. And once you understand each and every bacteria, the concept behind each and every bacteria, that chart will become super easy. Okay, just give some time. That's the most important thing. Now see, we are going to finish microbiology in next 3 weeks. But does it mean that you're going to master microbiology in the 3 weeks? No. You are going to learn very slow only. Anyone. Okay, depending upon how much exposure of the subjects you have got, you'll be getting that pace only initially. But the key is to improve every day. Small improvements every day. Try to build up the habit of learning, which I'm going to teach you. Okay, different researchers say that any habits are built up in 66 days, and which has got 3 phases of 22 days each. So including the habit of learning, it is only built up. It doesn't happen all of a sudden. We see many students, they come to the academy, they're very enthusiastic, they want to learn, but after some time, they realize that, oh my God, I'm not able to learn, I'm not able to understand. So what they do is, they stop learning. It's not for me. They don't give that time. They don't give that time to develop their habit of learning. Initially, you might be able to study only 2 hours. Though we recommend the students who's joining Dental Amaze Academy, you should join Dental Amaze Academy only and only if you can spare 5 hours, not learn 5 hours. Okay, don't mistake us. We have to spare 5 hours. Maybe you will be able to initially learn only 2 hours. You have to build up that habit to 5 hours, 6 hours, and then slowly make it 7 hours, then 8 hours, then 9 hours, then 10 hours. It's very much possible. Only thing is you have to give some time for that, and you have to have some patience for that. Okay, that's the key. Okay, fine. So have patience, learn slowly, increase your speed. Once you catch the speed, put your full efforts and go at the fastest pace. Okay, so these are some basic knowledge about Staph aureus. Now, yes, the first topic, don't worry, don't read this. Students have got a very bad habit of reading what is there on the board and not listening to what is being said. Okay, we'll go one by one, don't worry. So the first topic I'm going to teach you is the resistance developed to antibiotics by Staphylococci. Now, what is the importance of this? The importance of resistance developed by this particular organism is that recently it has been noted that many Staphylococci are developing resistance to antibiotics very fast. You give one antibiotic, next time the patient comes with same infection or the infection by the same organism, that antibiotic is not effective, and you have to keep on raising. And finally, this stage reached where no antibiotics are effective. So you have to learn about the resistance developed in these organisms, and the most significant thing is that they develop serious resistance compared to streptococci even. Okay, they develop more resistance. Now, we know streptococci is very important. Isn't streptococcus mutants that is involved in dental caries? So you have been told, I'm sure, many dental students have this complaint that teachers tell that this is not important for you. Staphylococcus is not important for you, or this particular topic is not important for you. Cardiovascular system is not important for you. ECG is not important for you. And by the time you reach we feel, okay, yeah, true. Basically it's teeth. So why do you need to learn about heart? Okay, but by the time the student reaches final year, they start realizing that all these were so important. And unfortunately, I have seen people who can't even manage a medical emergency when it occurs in simplest of medical emergencies. They don't know what to do. Okay, they don't know what to do. So the problem is it's not being taught from beginning. Now, what is the importance of Staphylococcus for a dentist? It's a very common organism in dental caries. No. Dental caries, it is there, but more important is Streptococcus, definitely. But peri-implantitis, there is a disease which is a new disease, okay, which was not there before, as there was no implant before, but now Staphylococcus is being found all around the implants. So it is a very important organism. It's also involved in dental caries. Okay, you can see it in the oral cavity, and more importantly, this organism can cause endocarditis, inflammation of the endocardium. Okay, and what's the problem with that? When it comes from the oral cavity, after tooth extractions. So they don't know, the person who's going to teach you doesn't know initially, they don't know is there any connection between teeth and heart? Yes, they don't know this. So from now onwards, you should know the significance of what you're learning. Yes, you are here to get a NEET MDS seat, so concentrate on that. But whatever knowledge you get, we see many students, they think that I want to get all the knowledge in the world because I'm going to write NEET exam. Now, a very important question asked by one of my seniors long back when I was starting to prepare for my PG. The senior asked me, do you want PG or do you want knowledge? Because I was running behind knowledge. I said, I want PG, so don't run behind knowledge. Once you get your PG, you improve your knowledge. As of now, improve your MCQ skills. Definitely you need to learn systematically, you need to learn basics, but yes, not for knowledge, but to clear NEET MDS exam. Okay, so your focus should be clearing NEET MDS exam. There will be lots of things. When you're studying 20 subjects in the next 10 to 12 months, there will be a lot of things which you can't make logic upon. Okay, at times, majority, yes, there will be, but certain things you have to by heart. Because you're studying 20 subjects, we are teaching you 20 subjects in 12 months. So all the topics cannot be explained in detail. Okay, definitely as much as needed will be there. But you have to build up the habit of gaining or memorizing things out. And memorizing, please note, for NEET MDS comes only by revision. Okay, it is not sitting and learning one particular thing in one day. No, it comes with revision, which I'll tell you in the coming lecture. So coming back, so resistance to antibiotics, it is based or discussed under 5 headings, of which 3 are very important. 2 is, yes, emerging, the current understanding. So one is the secretion of beta-lactam. Second is alteration of target site. You will understand all this. Third is tolerance to penicillin. Fourth is VRSA and VISA. So what are they? We'll discuss one by one. Okay. So the first thing, beta-lactamase mediated. Now what is beta-lactamase? To understand that, you have to understand what is beta-lactam antibiotic. So you have this particular organism. Okay, bacteria, which has got cell wall. Okay, and there is a group of antibiotics known as beta-lactam antibiotics, which has got a beta-lactam ring, which we will study in pharmacology. The role of those antibiotic is to act upon this cell wall and destroy the cell wall. And once the cell wall is destroyed, the bacteria cannot survive. Okay. Now, what does these organisms do? They produce something known as beta-lactamase. So that will destroy. So imagine this is a beta-lactam ring. Okay, beta-lactam ring. So this beta-lactamase comes and destroys this beta-lactam ring. So you give the antibiotic to the patient. One of the example of beta-lactam antibiotic is penicillin. So you give penicillin to the patient, it is not effective because the patient is producing beta-lactamase, or it is also producing penicillinase. Now what is the difference between penicillinase and beta-lactamase? Now, for that you have to understand what are the beta-lactam antibiotics. The different beta-lactam antibiotics are, one is penicillin, other is cephalosporins. So if beta-lactamase is produced, it can destroy any beta-lactam ring, including cephalosporin. If the bacteria produces penicillinase, it can destroy only penicillin ring, okay, which is also a beta-lactam ring, but it can only destroy penicillin ring. Okay, so that is the 2 types of beta-lactamases in a broad sense. Now, this is plasmid mediated. What does it mean? This production of beta-lactam ring is plasmid mediated. Again, it comes under basic bacteriology, so I'll explain it here. Basically, if you consider a bacteria, it has got definitely chromosome. It will have a chromosome, okay, like, or I would say, okay, they have got circular chromosomes, isn't it? Bacteria has got circular chromosome. So this is a circular chromosome, and inside that chromosome there is DNA. This is a basic thing. Inside chromosome there is DNA, okay, in the organisms. Now, bacteria also has got extra chromosomal DNA. Okay, extra chromosome. It is a piece of DNA which is outside the chromosome, and this is known as plasmid. This extra chromosomal DNA is known as plasmid. Okay, now how is this beta-lactam, the capability to produce beta-lactam is transferred from one bacteria through the other by transferring the plasmid. So you have got a second bacteria here. What it does is it gives its plasmid to the next bacteria. Means the next bacteria can also produce beta-lactam. So that means that also becomes resistant to a particular antibiotic. Okay, fine. Now, what are the methods of transfer of this plasmid? There are two methods. One is transduction. Okay, transduction. And the second is conjugation. The second method is conjugation. So what are these methods? We'll go on to details of this in basic bacteriology, but just for you to understand. In transduction, you have got two bacteria. Okay, what it does is, okay, inside this bacteria there is that plasmid. Okay, and what the first bacteria does is it gives that plasmid to a vector, which is a virus. So the name of that vector, that virus, is bacteriophage. Okay, so it's a virus, bacteriophage, which depends upon the bacteria. So this virus takes in the plasmid, okay, and then it enters into the body of next bacteria, and then it injects or it puts in the plasmid here. So this method of transfer of genetic material, including plasmid, from one bacteria to the other bacteria through a medium of a vector or a bacteriophage, which is a virus, is known as transduction. So this is one way of transferring the capability of producing the beta-lactam. Second is conjugation, which is very simple. You have got two bacteria. Okay, again they have got a plasmid. Okay, what happens is there is a connection developed between these two bacteria. It can be something known as sex pili, which is an appendage. Okay, connection, a tube, okay, which develops, and through this tube, very simple, this gets inside this. Okay, so it's not 3-4, it's 1. Okay, so this is the method through a conjugation. So it's kind of joining. So there's a joining through sex pili, and this is the two methods. Okay, transduction and conjugation. So these are what you have to know. The first kind of resistance developed either by beta-lactamase or penicillinase. Okay, beta-lactamase or penicillinase, and it is plasmid mediated. Very important. They can ask you, beta-lactamase resistance is chromosomal DNA mediated or plasmid mediated? Plasmid mediated. Is it by which methods? Transduction and conjugation. Got it? So if you find a bacteria which has developed resistance to the beta-lactam antibiotics, you find that the normal antibiotic which you give for Staphylococcus aureus, the most advised antibiotic for Staphylococcus aureus is benzyl penicillin. Okay, Staph aureus, it is benzyl penicillin, but the organism has developed penicillinase. So what will you do? You have to give penicillinase resistant antibiotic. Okay, you have to give penicillinase resistant antibiotic, which is cloxacillin. All these we'll go and study in detail when we study pharmacology. Okay, just now know this. And dicloxacillin. Okay, dicloxacillin. Now, instead, if it has developed beta-lactamase, which can destroy any beta-lactam ring, then you have to give clavulanic acid. Okay, along with amoxicillin, you give clavulanic acid. Okay, so these are the methods. The first method, which was beta-lactam or beta-lactamase production. Okay, now we go to the second method, which is the alteration of target site. Now, what is a target site? We studied there is the organism, okay, and there is the cell wall. So on the cell wall, for the penicillins to act, there are some target proteins. Okay, there are some target proteins. Now, imagine the bacteria is changing the target protein. Okay, the bacteria is changing the target protein. Now penicillin comes, penicillin is not able to understand, because it can only recognize a typical kind of target protein, not all. So if the bacteria changes the target site itself, penicillins cannot recognize the site, and so they cannot act. Okay. Now there is a particular penicillin binding protein. The name of that protein is PBP2A. Okay, PBP2A. Just know this. Okay, PBP2A. Know the name of this protein. Okay, which gets changed. Okay. Now the organism, what we get as a result of changing of this particular target site is known as MRSA. Very, very important. Maybe many of you might have heard of this. Methicillin resistant Staphylococcus aureus. I studied previously that you can give cloxacillin, dicloxacillin, methicillin also, sorry. Right here, methicillin. Okay, methicillin, cloxacillin, dicloxacillin. So now, due to this particular mechanism of changing the target site, the organism has become resistant to methicillin. You can't give methicillin also. So that's a big problem. And as a result, all beta-lactam, not just methicillin, it has been noted that all beta-lactam antibiotics, okay, all beta-lactam antibiotics are resistant to MRSA. Plus, it doesn't stop there. Even the commonly used antibiotics like tetracyclines, then even cephalosporins, definitely it has to be, okay, cephalosporins, erythromycin, and aminoglycoside. So all this you will study, just write it down, you will study this in pharmacology. So even these antibiotics become resistant, okay, not just the beta-lactam antibiotics. So what is the solution? If a patient or before that, it's regulated by chromosomal DNA. We studied the beta-lactamase is regulated by extrachromosomal, which is plasmid. This is regulated by chromosomal DNA, and there is a specific gene here, SCC-MEC gene. Okay, the specific gene involved here, which is known as SCC-MEC gene. The full form of SCC-MEC gene is Staphylococcal cassette chromosomal MEC gene. Okay, chromosomal MEC gene. Okay, the name of the gene is SCC-MEC gene, which is being transferred. Okay, you have to very clearly, very specifically know the name of this gene, SCC-MEC gene. So that is being transferred. Okay, now as a result, it's being noted that there is two types of MRSA infections developed. Okay, there are two types of MRSA infections developed, and of which one is the hospital acquired MRSA, and the second is the community acquired MRSA. Now, why is I'm saying into so much detail? Because the MEC genes are different. We said that there are MEC genes involved. So in hospital acquired, this is authorized short forms, HAMRSA and CAMRSA. They can even ask you what is CAMRSA. It is community acquired MRSA. What are genes involved in the HAMRSA? It is the MEC gene type 1, 2 and 3. The MEC gene type 1, 2, 3. Okay, MEC gene type 1, type 2 and type 3, whereas here it is type 4. Okay, just no. And please note that this hospital acquired is much more dangerous. Why? Because it is multi-drug resistant. Many drugs, due to this particular mutation, many drugs become resistant. Here it is maybe a few drugs are resistant. Okay. Now, alternative, what's your option if a patient develops MRSA? The drug of choice, this is another very important MCQ. The drug of choice is vancomycin. Or you can also give teicoplanin, but vancomycin is the most important, is drug for MRSA patients. Okay, that's the second type. So we studied two. What are they? One is beta-lactam. Second is, so beta-lactam is plasmid mediated. Then we came on to the second type where the target protein is altered, PBP2A, and it is chromosomal DNA mediated. Okay. Now the next mechanism by which they develop resistance is by tolerance to penicillin. They start tolerating penicillin. That is, the organism is only inhibited and not killed. Okay. The penicillin is a bactericidal antibiotic. It kills the bacteria. But if this kind of a tolerance develops, the bacteria is not killed, it is only inhibited. What is the reason behind that? As of now, just know it is because of huge difference between MIC and MBC. We'll study this in pharmacology in detail. Minimum inhibitory concentration, minimum bactericidal concentration. There's a huge difference or huge gap between these two. Okay, as a result, the amount, if you're not understanding, forget it, but as a result of huge difference developing between MIC and MBC, the amount required to kill the bacteria will not be enough, however amount you're giving. Even if you give a huge amount of antibiotic, the inhibitory concentration is not reached. Sorry, the bactericidal concentration is not reached. Still it is in an inhibitory range because there's a huge gap developed between these two. Okay, as of now just know it. We'll go into detail of MIC and MBC in pharmacology. Then comes the Vrsa, vancomycin. We studied the drug of choice for MRSA is vancomycin, anticoplanin. Now imagine the bacteria develops resistance to that vancomycin itself. It's a big problem, isn't it? So what is the reason? One is MEC gene, which we know. There's another gene also involved, VAN-A gene. So it's a chromosomal mode of inheritance, isn't it? So chromosomal DNA. VAN-A and MEC gene, just know these two names, VAN-A and MEC gene. So if it's very important that if a patient or if a bacteria, you find that a patient is resistant, in fact a patient is resistant, isn't it? So if a patient is resistant to vancomycin, what's the drug you give them? You give linezolid. Okay, we'll study again, we'll study all this, don't worry. Okay, we'll study all this in pharmacology, what are the mechanisms and everything, but just now note it down. Okay, Vrsa, linezolid. Now, the next is VISA, that is vancomycin intermediately resistant Staphylococcus aureus. Intermediate, I is for intermediate. So, means it is not a proper resistance, it's a kind of intermediate resistance. Means there is in fact a decreased susceptibility. It is not pure resistance. It is not resistance. It is just the bacteria is not susceptible much. You give initially, yes, you find there is some effect, but then susceptibility comes down. Okay, and this is because of a thickening of cell wall. Cell wall becomes thickened, and not due to any problem with gene, not gene. Okay, there is no gene transfer or anything like what we saw previously. It is just a thickening of cell wall. So they can ask you, visa is due to thickening of cell wall. Okay, visa is due to the thickening of cell wall. And what is seen? Is resistance seen? No, it is decreased susceptibility to vancomycin is seen. Okay. Now coming on to the disease part of Staph aureus, which is pathogenicity. There are two types of diseases caused by Staph aureus. One is infections, staph infections. Second is intoxication. So the mechanism is different in both. In infections, the host defense is being compromised. The bacteria gets into the body and it will be playing with the defense mechanisms of the body. Okay, and the second type, it is intoxication, that is toxins are produced by this organism and these toxins will act. The bacteria will not do anything, the toxins will act. Okay, the toxins will be acting. So let us now see the virulence factors, which includes toxins. So there are three broad classifications or three broad types of virulence factors. One is cell associated, means inside the cell. Okay, inside the cell there is something. Okay, we'll see what they are, or maybe just attached to a cell there is something more importantly. Okay, cell or cell wall. Then we have got extracellular, something outside, maybe enzymes. Okay, they secrete some enzymes to out, and these enzymes play a role. The third is toxins, much more dangerous toxins. Okay, toxins are coming out. So three types: cell associated, extracellular, and toxins. Okay, so let us see one by one what are they. So the first type is cell associated, and you know outside the cell you have got a cell wall. So if you consider this as a cell, you have got a cell wall outside that. And please note, gram-positive cell walls are composed of two very important components, which is peptidoglycan and teichoic acid. So these two are virulence factors of this bacteria. It has got peptidoglycan and teichoic acid. Okay, and outside these peptidoglycan and teichoic acid, there is another layer which is the capsule. Okay, capsule. Okay, so this is another virulence factor, and there are some proteins on the cell surface. So just besides all these, you have got some proteins on the cell surface. So this is a cell surface. You have got cell surface proteins. So these are the virulence factors. So one is outside that cell surface proteins. Then you have got peptidoglycan and teichoic acid, a component of cell walls. Then you have got capsule outside that. So each of these has got some specific functions, of which the first one is peptidoglycan. What it does is it provides rigidity to the cell and shape of the cell. So rigidity is a very direct, simple things, rigidity and shapes. And also it activates cytokines. Now this is a huge topic of interest, especially in a COVID era. You would have heard of patients dying of cytokine storm, or maybe you might not have heard of cytokine storm. You might have heard of pneumonia, patient dying due to pneumonia, or fibrosis developed, isn't it? Or ARDS. All this, the key factor involved is cytokine storm. Cytokines are inflammatory mediators produced by our own body. It's not produced by bacteria. Our body produces it. But who signals the production? The organism. In case of COVID, the COVID virus signals the production of cytokines. Here, the peptidoglycan signals the production. So the bacteria goes inside the body, and it tells the body, produce this peptidoglycan of the bacteria will tell the body, produce cytokine. The body will think that, okay, I'll produce cytokine. To some extent, it is very good. It is killing the bacteria and all, but when it reaches beyond a level, cytokines starts harming the body. We'll study this when we come on to general pathology topics. Okay, what is the role of cytokines? Anyway, so see, why is it each and every, because it's just spread out, isn't it? Cytokines are here, maybe some topics are here, some topics are there. Now, why is it important to study NEET MDS for NEET MDS like this way? The only reason is to cover subjects. Okay, you can, like, right away from here you can jump to cytokines and I can teach you cytokines. But the problem is, the MCQ book you have, Dental Pulse or Dentist or any book, the thing is that the questions are arranged based on subjects. You have to cover the subjects. Okay. Also, it gives you an idea that how much subjects have you covered. That's the key. So you don't. One student once asked me, why can't we do all the related topics together? You will not reach anywhere. Yes, it will look very good, it will look wonderful, you will have lots of knowledge again, but it will not make you prepare for NEET MDS exam or AIIMS MDS exam. So rigidity and production of cytokines. Teichoic acid, very important. What's the role of teichoic acid? Teichoic acid is it's known as major antigenic determinant. So what is antigen? Antigen is something which helps the production of antibody. So and it's a part of mostly the pathologic organism. So teichoic acid is a major antigenic determinant. This is a question for you. Which of the following is a major antigenic determinant of Staphylococci? It is this teichoic acid, and also it facilitates adhesion. It helps in adhesion of this organism onto the surface of the host cell. Then only it will start attacking it, isn't it? So this happens. Now capsule prevents opsonization. Just know now. This is what I said. Microbiology is a very interesting subject. We'll study about opsonization, we'll study about phagocytosis, what happens and all. So opsonization basically, you know what is phagocytosis? Engulfing. A bacteria comes, macrophage of our body comes, engulf it, eat it. Now, to eat something, you need it to be tasty, isn't it? You need it to be tasty. So, for macrophage to eat a bacteria, it should feel like it's tasty. That increasing or putting some masala and increasing some taste is known as, or increasing the taste of the bacteria is known as opsonization. In simple terms, okay, opsonization. Simply, opsonins, certain molecules come and attach to, okay, we'll study what are opsonins. Opsonins come and attach to the bacteria. So once these opsonins come and attach to the bacteria, just imagine this is an opsonin. Macrophage now recognizes this bacteria. Otherwise, it will not recognize the bacteria. So it's a kind of making it tasty. Capsule, what it does, it prevents opsonization. It will not allow it to be recognized. Okay, very beautiful, isn't it? It prevents opsonization. But our body is more smarter. It has got different mechanisms. It come with complement. Okay, it will come with complement and kill it. So we'll study them. Now, and finally, cell surface protein. So, what are they? Peptidoglycan, teichoic acid, both of them component of cell wall, capsule outside the cell wall, and cell surface proteins, which we'll study next. Okay, so peptidoglycan very importantly in cytokines production and cell rigidity. Teichoic acid is a major antigenic determinant and helps in adhesion. Capsule prevents opsonization. Cell surface proteins is the next topic of interest. Now, there are two very important cell surface proteins. One is protein A, the other is clumping factor. Now, to understand protein A, I need you to understand how body attacks a bacteria. First, it calls, come here. Okay, tells the bacteria, the body has, or the inflammatory cells, especially the neutrophils in the body, tells bacteria, come here. It will call. Okay, and it will also call macrophages. Neutrophils, and it will say macrophages jump on it and kill it or engulf it. Even neutrophils, if it is small neutrophils, they also attack and engulf it. Now, if this doesn't happen, if this doesn't work out, it removes a kind of knife, kills the bacteria, and this knife, the killing knife, is known as complement. We'll study this in detail when we come on to the further part of immunity. So these are the mechanisms. It calls, engulfs. If it doesn't work, take the knife, stab. The protein A can resist all the three. The first, which is calling the bacteria, is known as chemotaxis. Okay, chemotaxis. Okay, chemotaxis. So this chemotaxis will not work. It is anti-chemotactic. Anti-chemotactic, protein A. Okay, and it is anti-phagocytic also. Anti-phagocytic, and it is also anti-complementary. Anti-complementary. Now, are they really important? Not much. What is the most important thing which you have to know, you must know about the protein A? It is, this protein A is responsible for a reaction known as coagglutination. Coagglutination, just write it down. It's very, 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 very important. It's responsible for coagglutination. Now, what is coagglutination? Coagglutination is a reaction which is done outside for recognizing this protein A. Do this particular bacteria, you don't know what bacteria is it, you want to check if there is protein A present. So if protein A is present, it belongs to this particular group of bacteria. So for that, you take or you undertake this reaction which is known as coagglutination. We'll study in detail about this in antigen-antibody reaction. So coagglutination is a type of antigen-antibody reaction. So where is the antigen, where is the antibody? That's what I'm going to tell you now. You take the bacteria, okay, and on surface of the bacteria, you have got the protein A. Okay, you've got the protein A. What you do is, you react this protein A with IgG. Okay, IgG. Okay, now you get antigen-antibody complex. Antigen is protein A and antibody is IgG. Okay, now you have a complex. Now, you put from outside a known antigen which you know. Okay, you know a particular antigen which can react with this complex. You want to find if this complex is there, this antigen will react and they form an agglutination, or they form a kind of clot. Okay, just think it this way. So if this protein A is present, this reaction will take place and you get coagglutination. If you don't understand anything, know that protein A is responsible for coagglutination. For your NEET MDS purpose, it's all over. Okay, for AIMS, you have to know this also. Okay, because they will tell you, in AIMS, they might ask you, a patient came to your clinic, okay, with this and this particular kind of symptoms and all those, okay, and when his serum was taken out and reacted with IgG, okay, it formed a complex which showed type of fluorescence. Then it was reacted with another antigen, known antigen, and it formed a coagglutination product. Coagglutination, the term came there. So they'll ask you, what is the organism, or what is which virulence factor is responsible? It is protein A. So AIMS, they will ask you the same thing in a twisted way. NEET, they will directly, most of the time, not always, they'll ask you directly. Okay. Now the second cell surface protein is coagulase. Have you heard of it? There are two types of coagulase, bound and free coagulase. We'll study them, of which the one which is on the cell surface is the bound coagulase. The other name for it is clumping factor or bound coagulase. Okay, bound coagulase. We'll study them in detail when we study coagulase. Okay. So as of now, just know cell surface proteins, protein A and clumping factor. Protein A is responsible for coagglutination. Clumping factor is coagulase B or bound coagulase. Okay, coagulase B or bound coagulase. Now, that was the first type, that is cell surface components. Now, extracellular, what comes out, and majorly they are enzymes. One is coagulase, of which it is the free coagulase which is an enzyme. Bound coagulase is a cell surface protein. Remember that. You shouldn't confuse. Though there are two types of coagulates, one is free and one is bound. The bound is a cell surface protein which is attached to the cell, and the free coagulase is a enzyme which is free. Okay, and then you have hyaluronidase. Now, I'll discuss coagulase in detail, but as of now, I want you to understand something known as hyaluronidase. The other name of this hyaluronidase is staphylokinase. Staphylokinase. Now, why staphylo? Because remember, later we'll study, this hyaluronidase is also produced. This hyaluronidase is also produced by Streptococcus as well. So this is staphylokinase or fibrinolysin. The other name is fibrinolysin. Okay, fibrinolysin. So what is the role of it? It's very important to note that this hyaluronidase or this staphylokinase breaks down the connective tissue. You have got, if you just take your cheek Okay, there is a connective tissue. So bacteria, this connective tissue is a big hindrance for the spread of the bacteria. So this hyaluronidase can break that connective tissue septa and go through it and spread. So this is important in the spread of infection. Now, which organism produces more hyaluronidase? Is it Staphylococcus aureus or is it Streptococcus? It is Streptococcus, not Staphylo. Staphylo is usually localized infections, except a few which can produce this hyaluronidase. But Streptococcus produce more hyaluronidase, and thus it is more kind of a generalized spreading infection. Note that staph, though they produce this hyaluronidase or fibrinolizin, much easier to pronounce, fibrinolizin, it is less kind of a spreading infection. Okay, they can ask you that also. Which produces more spreading infection and why? Okay, it's Streptococcus which is producing more spreading infections. So now let us come on to the coagulase. I told you the type A is the free form, which is the extracellular enzyme. Type B is the bound or clumping factor, which is cell surface protein. Don't confuse that. Though I'm discussing it under extracellular enzymes, don't confuse it, because that's why I discussed it, the clumping factor before itself. Now, as I've told you, there are two, type A and type B. Type A is free, type 2 is bound, known as clumping factor. Please note that the type A is heat labile. They can ask you all of the following are true about the type A coagulase. Remember, it is heat labile. I'll tell you how to remember it. Okay, and type B is heat stable. Heat stable. Now, the next thing which you have to know here is this forms the basis for tube coagulase test, and this forms the basis for slide coagulase test. Slide. So you have a tube, okay, you have a test tube where you have a free coagulase floating from top to bottom, okay, and you know, just try, if you don't know, heating a test tube will burst the test tube. That means it is heat labile. So that's how you can remember it. This is a free coagulase, tube coagulase test, and heat labile. It will burst. Okay, the exact opposite, slide. So slide is much more resistant to, you know, the slide is much more resistant to heat. Okay, and it forms the basis for slide coagulase test, and it's heat stable, and it's the bound coagulase or the clumping factor. So let us go into a little bit detail. Though if you know this much, it is enough. But I'll just tell you, because at times they will give you application level question based on they're conducting a coagulase test. Okay, so let us see into that. So that is the tube coagulase test is what I'm going to discuss. The tube, the test tube, that is the free coagulase. So what you're going to do is you take a test tube, you have the Staphylococcus aureus culture which contains a free coagulase. Basically, where is the free coagulase coming from? It is from the staph culture, isn't it? So that is the agar culture which we'll study later on. Okay, and you mix it with rabbit plasma, the plasma of the rabbit. Mixing with human plasma doesn't work out much. So what happens is that this mixing will activate CRP. What is CRP? C-reactive protein. Huh? No, it is not C-reactive protein. C-reactive protein also CRP, but this CRP is something different. The CRP, what we have been knowing, which is C-reactive protein, is an inflammatory mediator, okay, which is important in COVID as well. This is coagulase reacting protein. You get a question, which of the following type of coagulase test involves the activation of coagulase reacting protein? It is the tube coagulase test. I will tell you in detail, you understand it. Okay, so this combination leads to the activation of the presence of free coagulase in rabbit plasma leads to the activation of CRP. Now what happens? You get a complex, okay, you get a complex of CRP and coagulase or free coagulase to be very specific. Now, what is the role of what happens if the CRP coagulase complex is formed? Note that this complex can activate prothrombin. And once this prothrombin is activated, coagulation. Okay, what is the steps in coagulation? Prothrombin can convert what? Prothrombin can convert fibrinogen to fibrin. Fibrinogen to fibrin. And this is what is the plasma clot. This is known as positive coagulase test. Plasma clot, that is positive. The test is positive. Now, how can you get a question here? Patient came, all those big details will be there. Culture was done, and the culture was mixed with rabbit plasma, and there was a plasma clot noticed. They will not tell you there is CRP. There was a plasma clot noticed, means there is coagulase test positive. And what coagulase test was done here? It is the tube coagulase. Okay? In bound coagulase, you can get it negative even for positive organisms. So tube coagulase test is much more efficient than the slide coagulase. Okay, so here note that there is the involvement of CRP. Okay, there is the involvement of CRP there. Okay, and you get a plasma clot. Okay, so basically positive coagulase test means nothing but plasma clot formed. Now let us see the bound coagulase. What is the difference? Or the slide coagulase test. Here, the bound coagulase, which is cell surface protein, which is there on the cell wall, okay, which is projecting outside the cell wall, or which is there on the cell wall, cell surface, okay, it's a part of cell wall, okay, and this reacts, there is no CRP activation, nothing, it directly reacts with fibrinogen. There is no involvement of CRP, and you see, because it's on a cell wall, you see the bacterial cell clumping. That's why it's called a clumping factor. Okay, you simply see that fibrinogen just deposits onto the bacterial cell wall, and thus there is a bacterial cell clumping. Simple. So there is no CRP. That's what I want to tell you. The key, what I want to tell you here is, there is the no involvement of CRP, and the type of reaction is different. This bacterial clumping formation, okay, and here it is clot formation. Okay, now on your screen you can see the two different types of coagulase tests, and coagulase positivity and coagulase negativity. On my left side you can see the slide coagulase test, on my right side you can see the tube coagulase test. So the slide, you get the clot. You can see there is positive clot formation, isn't it? So that is a kind of crumbled amalgam, okay, this clot formation. On my right side there is the tube coagulase test, and you can see that clumping has happened in the positive tube. So you can get images on this, okay, just know. And it's one is slide and the other is tube, just know it. And now, is it important to read textbook? Many students ask this. Is it important to read textbook? Textbook reading has to be limited. The problem with reading too much of textbook is that, see, I told you, learning comes only with revision, isn't it? How many times can you revise your standard textbook if you are following your textbook? It's very difficult. So two things I want to tell you here. Write down your notes. Limit your reading of textbook. What is the advantage of that? What is the advantage of writing down your notes? What is it? The advantage of writing down the notes is that your own notes, it's not instead of being printed notes, okay? Write it down. Yeah, academy will give you soft copy notes, hard copy notes, all those, but I would suggest, I've seen many toppers, everyone, they are writing down their notes. What is the importance of, what's the use of writing down the notes? The key there is, you are writing it, you are listening it, you are seeing it. That means multiple senses are working and you are writing it in your own handwriting. What's the advantage of doing that? The advantage is you can learn faster or you can revise faster. Okay, if you're someone who have written down all this so far, Just take the next 10 minutes and go through it. It will be so easy for you to learn. Instead, take your textbook and start reading. The same thing, same topic. Okay? And if you start reading it, it will take you the whole day. But if you have even solving your MCQs from your dental pulse or any printed notes, it will take time. But if you write down your notes, the best part is that it becomes super easy for you to learn. Secondly, you will reach a phase in your final round, final revision, stage 6 revision. Okay, stage 6 revision, your final revision before 2 months before the exam. So stage 1, 2, 3, 4, 5 will take place as you go on. Okay, so in your stage 6 revision, you will realize that, you will notice that you have to go very fast, maybe 4 subjects in a day. So how can you finish 4 subjects in a day if you don't have your own handwritten notes? And please don't mix lecture notes with any of your notes. You might be writing the entire textbook. Even after saying multiple times, still students will start writing the entire textbooks. Okay, they are preparing notes, but now you are free. You are studying only 1 subject at a time. A stage will come when you are going to study 2 subjects, 3 subjects, 4 subjects, not at a time. You'll be revising them. So now you have completely free the first chapter in your entrance preparation. You have the whole day, like the student asked me. But at a later stage, it becomes very difficult to go back to all the contents and do it again. So this is where your notes are going to play a role. And as I told you, don't mix your lecture notes with any other notes, because this is the core area which you will be revising in your last round. Just the previous NEET, many students told that so many questions came from our notes. The only reason is it's a core area. We are teaching you the core area. So if you are able to confidently answer them, these core area questions, you are there with a very high chance. Okay. Plus you are learning more MCQs, you are solving more MCQs, learning your dental pulse, you are learning other MCQ books, all those. So that's will be more than sufficient. Now, what are the books to follow for your microbiology? I would suggest definitely one is Dental Pulse. Okay, and it is a must-have book. And please don't ask for PDF and get the latest edition of Dental Pulse, which is if you want, you can buy Dentist also. But I personally recommend Dental Pulse. Okay, because it's much more simplified version. Dentist is also a very good book, but you have choices, preferences. Different people have different preferences. Do you need any other book? Yeah, if you have enough time, if you think that you can spare a lot of time and a little bit extra money, then you can go for a book called Aravind Arora. Okay, it's a MCQ book. Okay, so if you want to solve more MCQs besides what you get from the academy, go for the Aravind Arora. You can solve it for bacteriology, viral infections, sterilization. Very important topic for AIMS. So all this extra is for AIMS. Okay. So if you can afford, even multiple students can combine and buy this book, Aravind Arora. Not Dental Pulse. Dental Pulse should be a personal book because you have to refer many subjects from there. Okay. So that's about the books to follow. Aravind Arora, if you can, if you want, don't hesitate to spend some money for something which is going to pay back you money of 50,000 plus as stipend. Now the third virulence factor we studied is toxins. And there are 5 toxins: hemolysins, leukocidins, enterotoxin, TSST, and exfoliative toxin. TSST is toxic shock syndrome toxin. So all this I'll discuss in the next session. Okay, so what you have to do now, just revise take some rest if you want, come back, not a long rest, 10 minutes rest, come back, revise in the next 10 to 20 minutes, revise whatever you have learned so far, okay, and done. Okay, that's enough. Wait for the next lecture, and once the next lecture comes, do it, and slowly by slowly start your dental pulse, then do the academy questions, and then go for the test papers and all those, and slowly by slowly, as I've told you, we will build up a a very strong foundation on top of which you can build anything, even Burj Khalifa also. Okay, so thank you so much. See you in the next session. Bye.